All right. Can everyone in the back hear me okay? All right, great. Thanks. Okay, thanks for that introduction. And there's the title of my talk. You've already heard it, Silent Majority, How Symbiotic Bacteria Evolve to Help and to Hurt. Let's make sure I can get this to work. All right. So my talk is going to have three pretty different parts, so I'll give you a quick introduction. Part one is going to be the longest part. It's going to be kind of a breakneck tour of symbiosis. So I won't just be telling you about the human symbiosis of the bacteria, but I'll be telling you about a lot of other symbioses as well. There's a couple reasons for this. One, it's interesting just to learn about biology of symbioses, but also what we'll take away from this is some general patterns. And these general patterns will be important for us. In the second part, we'll talk about a little bit of theory. One of the patterns from chapter 1 of this talk is that there are many symbionts that are beneficial and some that are harmful. In part 2, the theory will try to understand why this is the case. In part 3 of the talk, I'll tell you more specifically about some of my research that tries to understand how hosts can control their symbionts and whether we can do anything to select good symbionts and get rid of the bad guys. So we'll start with one of the main messages that I'd like you to take home tonight, that we are not alone. But first, let's begin with a couple definitions. First, symbiosis. Symbiosis simply means unlike organisms living together. It comes from the Greek, syn meaning with, biosis, of course, meaning life. But more specifically, the way I want you to think about it tonight is that symbioses are intimate associations between microbes and plant and animal hosts, and we'll be talking about both of those. So there's really two kinds of players we'll be thinking about. First, the symbionts, and these are microbes that live in some kind of chronic close association with a host. So when I say a chronic association, I mean it's something that you're carrying around with you for your whole life, not just like getting an infection and goes away. It's an infection that you have pretty much since soon after birth. And then, of course, the host, and these are the plants or the animals that become chronically infected with the microbes. All right, so that's our vocabulary lesson. Now, if you decide to fall asleep for the rest of the talk, here are the bullet points that I want you to take home right away. We are not alone. Well, what does this mean when I say we are not alone? Well, it means four main things. Point one, we are composed of multiple species communities. And this includes microbes that are difficult or sometimes impossible to separate from us. Two, we interact with the environment literally through layers of symbionts. Whenever we eat something, it's going through a layer of many different species of bacteria. Our skin is covered with many species of bacteria. It's basically something that we're constantly carrying around. Three, our symbionts range from harmful to helpful. This is going to be the main theme of the talk and something we're going to try to understand better. And four, in case you got an idea that maybe you can take some antibiotics and get rid of all your symbionts, I'll tell you right now that's essentially impossible. Some of our symbionts are required for our survival. All right, so if you want to go to sleep, you can go to sleep now. But if you want to stay awake, I'll give you a quick kind of breakneck tour of symbioses. I hope it's not too fast, but we'll kind of travel the world, and we'll travel it very quickly, and hopefully we'll come away with some facts about symbioses and some common themes. We'll start our tour at the Great Barrier Reef off the east coast of Australia. Probably a place that would be wonderful to go snorkeling or maybe scuba diving. I've never been there, but I'd love to go. And if we went there, one of the most amazing things that we would see are coral reefs, these incredibly diverse systems with all these very simple animals. Now, of course, the very simple animals are the corals. But if I was going there to snorkel or to see this beautiful system, one thing that I would be fascinated by would be thinking about what's going on inside these corals. These corals' tissues are literally completely infected with many different species of algae. And what these algae are, they're essentially like single-celled plants. So they're photosynthesizing, they're gathering energy from the sun, and they're giving some of that energy to the corals. And literally all the color of the corals that we see here is contributed from the algae. So if you took away the algae, the corals would have no color at all. So it's a nice cooperative system. The algae are giving their hosts sugars, and the hosts are giving some nutrients back to the algae. Everyone's happy. And the hosts need to be infected with the algae to undergo their normal life cycle. Now, what's particularly interesting about this symbiosis is that it's very vulnerable to breaking down. And if you've ever heard about what's happening in the coral reefs across the planet right now, something called coral bleaching. And what coral bleaching is the breakdown of this symbiosis. The coral, whenever the coral is at temperatures that are a little bit too high, or maybe UV irradiance that's too high for its symbionts, they lose their symbionts. So here we see one of the biggest bleaching events that's occurred in the Caribbean Sea. This is 1996. Nice healthy coral before. Here's after the bleaching event. You can see it's lost its color. Here's another bleaching event in the Caribbean. 2001, nice healthy reef. 2002, most of the symbionts have been lost. 2004, it almost looks like it's healed up again, but it's actually just covered up in slime, and most of those corals are dead. So this is a symbiosis that can easily break down. All right, some quick facts about coral-algal symbiosis, some things that we know, some general important aspects. One thing that's interesting is that when all corals start their life, they start their life without symbionts. So when they settle down in the bottom of the ocean and they are starting off life as a coral, they literally have to pick and choose their algae from the seawater. They have to find them and figure out which ones are the good guys. Fact 2, all these algal symbionts are infectious. So when we often think about infections, we think about infections as being a bad thing. For the coral, they have to get infected with these algae to undergo their normal life cycle. And once they become infected, they're literally spreading their algae back into the environment. So these algae can travel from host to host to host, kind of like an epidemic, but in this case, a beneficial one. Okay, fact 3, all these hosts have multiple infections. So they don't just have one kind of algae, they have many different kinds of algae. So it's kind of a complicated system. And finally, biologists have done all sorts of infection experiments where they'll take a coral and they'll infect it with different types of algae. And one general rule that they find is that although most of the algae are beneficial to the host, every so often they find some strains that are harmful, and they wonder, why is this the case? All right, so that ends part 1 of our tour. We'll leave the coral reef and we'll go to a beautiful spot, perhaps the Pacific Northwest or maybe just Northern California, going through some beautiful forest. But if I was hiking through this forest, what I would be thinking about is the incredible things that are happening below us in the soil. And below us in the soil are some incredible symbioses that all trees, and in fact most plants on Earth, have with many different diverse species of fungi. And these interactions are called mycorrhizae. And essentially what it is an interaction in which fungus is completely covering the roots of a tree. Here's a tiny seedling starting off life. Its roots are covered in fungi. And then there's actually some of these interactions which the fungi invade directly into the plant cell's tissue. These are called arbuscular mycorrhizae. And here we can see fungal tissue literally within plant cells. So just like the interaction between the corals and the algae, it's a beneficial interaction. The mycorrhizal fungi are usually giving resources to their plant. They're making the plant more efficient at picking up resources from the soil, and they're doing this in exchange for nutrients from the plant. Okay, so when we think about a tree starting off life, this can be difficult for a tree. So here's a nice tree to think about, the Scots pine. It lives in the highlands of Scotland, and it often lives in these big open habitats. And when that tree starts off life, it's kind of a big challenge. It's got to compete with all sorts of grasses and shrubs, and it's really got to dig down into the soil and get a lot of energy. So here's a tiny little Scots pine seedling. It's very cute. And we could look at its roots, and it looks like it's starting off life pretty good. It's got a good start. But if we took a more realistic look at that plant's roots, by taking away all the mycorrhizal fungi, we'd see how incredibly important the symbiosis is to the initial part of this plant's life. Here's what this tree would look like without its fungi. So when we're looking at a plant's roots, sometimes we might think we're just looking at a plant, but in fact what we're looking at is multiple species that are all interacting together. It's a much more complicated system than we would think. Now, if these fungi seem very unfamiliar to you, I'll show you the fruiting body of one. Of course, it's a mushroom that we might be very familiar with. So when we're walking through the forest, we don't have to look underneath the soil, but we can simply look at the mushrooms that we see around us. And most of those mushrooms are the fruiting bodies that are connected to mycorrhizal fungi that are, of course, having these very important symbioses with the trees that we're walking past. Okay, some important facts about mycorrhizal fungi and their symbioses. Fact 1, all these trees are born symbiont-free. So when that little seedling puts its roots down into the soil, it has to find its fungi. It's got to find its symbionts, and it's got to figure out who are the good guys, who are the bad guys, and it better pick the right partners. 2, fungal symbionts are infectious. So it sounds familiar from before. Those fungi can travel from tree to tree, from plant to plant, just like an epidemic, but in this case, beneficial. Point 3, all those trees have multiple infections. So again, when they reach down into the soil, they're not just picking out one fungus, but they're interacting with many different types. And also, just like we learned before, some of these infections, are beneficial, and some of them are harmful. And biologists have learned this by doing different infection experiments. So just like the biologists that are studying the corals, they wonder, why is this the case? Why are some of these symbionts bad guys? Okay, so if you wanted to learn about the human bacterial symbiosis, this is the part to really pay attention. Now we're going to delve into the human gut and look at some of the symbionts that we harbor. This is one of the most complicated systems. One of the things that's really problematic for this to study is that all these bacteria that are very important for our digestion are very concentrated in our distal gastrointestinal system, mostly our colon. And these bacteria probably have a lot of different kinds of functions, and biologists are just starting to unravel what these functions are. But we know that they're important for nutrition, important for breaking down nutrients that are unavailable to us otherwise. And we also know that they're important for the development of our immune system. Okay, some important facts to take home to think about our own symbiosis. One is, let's count the number of cells in the human body. If we were going to do this, a good estimate would be that there's about 10 trillion cells. So to give you an idea of how much that would be like taking all the humans on the planet right now and multiplying that number by about 2,000. All right? Then you have the number of human cells in the body. Now remember I said human cells. If we were going to count the bacterial cells that we carry around, it would be about 100 trillion. So in other words, just counting cells, we're only about 10% human, we're about 90% bacteria. Of course, you don't need to worry about it too much because these bacterial cells are very tiny. And if I could somehow take my body and break it up into different parts, I won't tell you how much of the part would be human, because then you'd know how much I weigh, but about 3 pounds of me would be all pure bacteria. Okay? Now, if we were going to try to count up the diversity of those bacteria, I'd be carrying around somewhere between 500 and 1,000 species of bacteria. And you notice there's a huge range in that estimate, and that just means that we just don't know that much about the diversity and how much it varies among people. Now, when the dean introduced me, he told you a little bit about the genes that we carry around. We know the human genome very well. Human genome has about 25,000 to 30,000 genes, depending on who you ask. If you were going to look at the genes of the bacteria that we carry around, it would be about 100-fold more. So every gene encodes a function. That function might be breaking down some protein or creating some important metabolic product. And what this tells us is that there's a great deal of function occurring, especially within our gastrointestinal system, that's not controlled by us. It's controlled by these hundreds of species of bacteria. Okay? So we know that these bacteria are digesting a lot of things that we cannot. They're providing important nutrients, immune benefit. But here's the bad news. There's all sorts of ways that the system can break down. There are many different ways that these bacteria can grow out of control. We can get infected with bad strains. There could be imbalance. And there's not much known about this, but we know more and more, there are more and more pathologies that are being racked up on this list that are directly related to our gut bacteria. Some of these pathologies include allergies. More and more allergies are thought to be closely related with our bacteria. Crohn's disease, diarrhea, gastroenteritis, meningitis. Obesity is probably tightly linked with problems with gut bacteria, pneumonia, septicemia, ulcers, urinary tract infections. The list could go on, but I won't continue. All right, back to some important facts about gut bacteria before we move on. 1, we are born bacteria-free. So when we are born, we literally have no bacteria at all. We are completely sterile, and that's why I've put a picture of this baby. This is actually my daughter. She's 8 weeks old in a couple days. So when she was born 2 months ago, she literally started bacteria-free, and she had to go through this process, just like the corals, just like the trees, of picking up her symbionts and hopefully making the right choices. Point 2, her bacteria, just like the other symbionts, are infectious. They can travel from host to host. Of course, we have multiple infections. As I've said, we can have many hundreds of species. And as you've also learned, some of these infections are good, and sometimes they're harmful. So these questions should start to seem kind of repetitive. All right, if you're getting bored, our tour is almost done. We're going to end our tour looking at some crops or maybe some wild species. It doesn't matter as long as you're thinking about beans, because that's what we're going to be talking about. And that's what I focus on in my research, because it's a very simple symbiosis that we can dissect and understand quite easily. What's fascinating about beans for me is that they harbor bacterial symbionts. Here's some of the bacteria in a plate that I've cultured. Here's a very tiny bean plant, a baby that's starting off life. It has to pick up its bacteria. And in these bean plants, we can see where the symbionts are. In the roots of the plant, they have these tiny little tumors. And those tumors are called nodules that carry bacteria. And what these bacteria are doing is they're literally fertilizing the plant. They're giving the plant nitrogen. Nitrogen is basically the most limiting resource for all plants other than water. And for these plants, they carry their own fertilizer systems with them that are these bacteria. All right, so here's a nice field. Actually, this is a place that I have done some research. We could zoom in on some plants, and we can see some of the bean plants that I study. This is a tiny coastal herb called in the genus Lotus that I study. And if we were going to dig one of those plants up and look at its roots, we could see some of the tumors, some of the nodules where the bacteria are. And here's a zoom up so we can see what these nodules look like. And they're literally plant organs that are created just to house these bacteria. So again, here are some of the strains that I've cultured. And just to remind you, these bacteria are incredibly beneficial. And we know this because we're always doing infection experiments. So here's a plant that's been grown up for just 8 weeks, and it was infected with a beneficial strain. Here's a plant that was also grown for 8 weeks, no bacteria. And this is a general rule. These bacteria can increase the growth of the plant by a great deal. All right, we'll finally finish up with some more facts. Legumes, of course, begin life symbiont-free. When they dig their roots down the soil, they have to find their bacteria. These bacteria, which are called rhizobia, are infectious. They can travel from plant to plant to plant. Bean plants are multiply infected. They carry many different strains. And as I've told you, although most of these infections are beneficial, some of them are harmful. All right? So you don't really need to take notes. What we're realizing is that although we've looked at these very different systems, we can start to take home some common facts. All right, so if you've fallen asleep and you've missed the whole first part, here's a quick rundown. Basically, anything that you look at in nature is infected. It's infected with bacteria. It's probably also infected with some fungi, maybe some algae too. Almost everything is born symbiont-free. There are definitely exceptions. Symbionts generally are infectious. So when we think of infections, we often think of this as a bad thing, but really, more often, we're infected by things that are good and are providing us important benefits. Almost all hosts have multiple infections, many different kinds of species, sometimes many strains of single species, and this general rule pervades that although most of these symbionts are beneficial, some of them are harmful, and this leaves us with the question of why is this? Why are some symbionts harmful? Why does this take place? All right, so this brings us to part 2 of the talk. If you were interested in natural history and a quick tour, you might find this part a little bit different because now we're going to talk about a little bit of theory. A little bit of theory about why do some symbionts help us while others hurt us. And this theory that we're going to talk about actually comes from medical research, and it's a type of theory called optimal virulence theory. All right, so this might be some unfamiliar words, so we'll go through a couple quick definitions. Virulence might sound like a familiar word, and what virulence is, it's a description of a trait of a pathogen. And it's a trait of how harmful is that pathogen? How much disease does it cause you? So now we've shifted to talk about bad infections, and we want to figure out how bad is an infection. Okay? So it's literally just the degree, it's the degree to which a pathogen harms its host. So this theory is literally a theory trying to understand how can virulence evolve? How can it change over time? So if we're thinking about a pathogen, how can it shift from being more or less harmful to a host? Now, if we were going to choose a pathogen to think about, we might choose a pathogen that has been on lots of our minds recently, certainly on my mind being a new parent, influenza. Of course, influenza is a virus, and sometimes it can have quite a bit of virulence. But what's interesting about influenza, what's interesting about the flu, is that there's quite a bit of range of virulence. Sometimes we might be infected with the flu, and we might have no symptoms. We might be asymptomatic, we get infected, that thing is dividing within us, doing its thing, going to a new host, and we never even notice it. So that would be harmless, that would be extremely low virulence. Of course, the very unfortunate part of recent flu epidemic that's occurring right now is that some of the strains, some of the infections have extremely high virulence. So one thing that medical researchers and also evolutionary biologists want to do is understand, why is this the case? Why are some infections very harmful? Why are some harmless? Can we make any predictions? And this is where optimal virulence theory comes in. All right, so here's the main idea. So now we're going to try to think about the world through the eye of a pathogen. A pathogen is making its strategies, and we want to think, what's the best strategy if I'm a pathogen? Now, the main thing a pathogen wants to do is it wants to make copies of itself and transmit to a new host. Transmissibility. So that's what we want to think of. How can I make copies of myself in the host that I'm in and get to the next host? All right, so here's the range. It's either going to be harmless or deadly. And what optimal virulence theory says is that most pathogens are going to be somewhere in the middle. But why is that? Now, if a pathogen is harmless, one way that a pathogen can do that is simply by dividing very slowly. Maybe it's just making very few copies within your body, and it's just not taking up many of your resources, not causing your immune system to do much. But the problem with that, if something is dividing so slowly that it's harmless, it's not going to be able to be very efficient at spreading to the new host. So if different pathogens are competing within your body, natural selection is probably going to favor the ones that are dividing a little bit faster, and those are the ones that are going to be better to spread. So it's going to be pushing it up the hill, saying, be more virulent. Now, if you think about that, the pathogens might become more and more and more virulent, but there's going to be a limit to that, because the pathogen needs to make sure it doesn't kill off its host too fast, because remember, that pathogen has to transmit to the next host. So a pathogen is always kind of waving this balance where they want to make sure that they're making enough copies to get to the next host, but they don't want to take over the system so fast that they kill you, because that's a dead end for them. They need to make sure that they jump ship in time. All right, so that's the first part of virulence theory. And if you're confused, I'll have to apologize, because it's about to get a little bit more complicated. That tells you the steady state for virulence, but what we really want to understand is what would make a pathogen shift to becoming more or less harmful to its host. What are the conditions under which that might occur? Now remember, I said the key thing to being a pathogen is being able to spread to new hosts. So a pathogen can afford to be more virulent, can afford to harm you more when it's easier for it to spread to new hosts. So these are some important conditions that can favor high virulence. Easy transmission, if it's easy to get to the new host. Of course, this can happen also if many new hosts are available. And finally, a very important condition is multiple infections. Within your body, when you have multiple different infections, different pathogens competing with each other, it's not just a lottery. The pathogen that's dividing the fastest, the one that's growing most rapidly, is often the one that's going to win the battle. So if you have multiple infections, that can cause the one that's most harmful to win out. There's also the opposite side of the spectrum, too. These are factors that can cause lower virulence, and most of them the opposite of what I've said before. Oops. If it's very difficult to find the next host, you better not kill the host that you're in. You better have lower virulence. Lower virulence is going to be favored. If hosts are rare, if you're in a single infection and you don't have to compete against other pathogens. And then finally, something that's very important, host response. This is essentially like the immune system. Your immune system is adapted to taking care of the pathogens that are going to be most harmful to us. So the immune system itself can literally select against the pathogens that are most harmful and select for lowered virulence. All right, so I've just bombarded you with a lot of facts, a lot of theory, actually. And what I can tell you is that almost all of these ideas have been pretty well supported by simple experiments in the lab. But the problem is that experiments in the lab don't always tell us whether these ideas of mathematicians and theoreticians are correct. So what I'll do is I'll tell you a couple stories about the evolution of virulence, some real stories, and we'll try to decide whether the evolutionary theory is supported or not. In the first story, we'll go back all the way to 1918, a very terrible world pandemic of influenza that occurred. It was called the Spanish flu. And the Spanish flu, which occurred during World War I, of course, it killed at least 50 million people. It killed a lot of young men, of course, that were serving in the armed forces. It may have killed as many as 100 million. The problem is that it was such a disaster that the numbers were not kept very well. Now, after this terrible world pandemic occurred, there were a lot of people that were really interested in trying to figure out why this happened. What was so terrible about this particular virus that spread across the world in 1918? What was so deadly about it? Because remember, influenza's been around for hundreds of years. What was so different about 1918? And a very interesting man named Johann Holton was particularly interested in this idea. And he was a graduate student in microbiology in the early 1950s at the University of Iowa. And he had this idea of going up and trying to excavate some bodies from 1918 and try to extract this virus. And his idea, it was pretty wise idea. He had the idea of going on an exhibition up into Alaska because this influenza virus, it was infecting people all over the place. So his idea was to dig up some native Alaskans from the permafrost, extract the virus from them, and then infect some mice and study this 1918 flu and really sort out, like, why was it so deadly? So he did that. He was in graduate school, and, well, it's not the greatest story. He failed. He never got it. He went up to Alaska. He exhumed the bodies. You can only imagine the paperwork to do something like that. He went back to Iowa with tissue, and he was never able to extract the virus. It was a failed mission. So he dropped out of graduate school and switched to something more practical. He went to medical school, became a pathologist. He had a very successful career. And almost 50 years later, he was living in California now in Marin County. He learned about another exhibition that was going up to Alaska, back to Brevig Mission, this tiny town of 276 people northwest of Nome. And they wanted to bring him along. And he went along on this mission. And this time, they extracted the viruses successfully. And they brought them back to labs. And of course, this is now the late 90s, so we have much better technology. And instead of trying to culture the virus, they sequenced it. They studied the DNA of the virus. And this took a while. But in 2005, the full sequence of the Spanish flu was assembled. And it was announced. And it was very exciting that they had literally re-extracted this virus, and now they could study it. All right. The bad news is that when they studied this virus that killed between 50 and 100 million people, there was nothing unique about it. There was nothing about it that looked particularly different from other variants. So they were wondering what was so bad about this virus. Now, one hypothesis about this is based purely on optimal virulence theory. This hypothesis says that in 1918, there was essentially a perfect storm. All these conditions that were completely set up to favor a strain that would be incredibly virulent and spread through the human population. Of course, humans were incredibly crowded. There were all these barracks where people lived. Transmission was extremely easy. Of course, new hosts were always available, favoring rapid spread of virulent strains. New men were always being shipped off to Europe. And of course, there were many opportunities for multiple infections as people were being shifted around, brought into hospitals, and people were often transmitting infections to each other. So this idea suggests that maybe there was nothing unique about that strain, but it was essentially the perfect storm to select for extremely high virulence. All right, so now let's do one more story and then we'll move on. This is a story with a very cute star. The star is the European rabbit. And this rabbit was so cute that a nice man who was living in Australia decided to introduce some of these European rabbits to his estate. And these rabbits did what most rabbits do. They multiplied. And over a relatively short span of time, these 24 rabbits, bred, and they spread across the whole continent of Australia. And soon, the populations were estimated to be nearly 1 billion. So these rabbits were running amok and causing all sorts of destruction across Australia. And here brings in the other part of the story, not nearly as cute, the myxoma virus. Now, some Australians planned in the 1950s to introduce the myxoma virus. This is a virus that they knew was deadly to rabbits, to control the rabbits. Basically, they wanted to eradicate them. And it was incredibly successful. It literally decimated the rabbits, killing 9 in 10. And over the span of several years, most of the rabbits were killed. But then something interesting happened. Over the next couple decades, the virulence of the pathogen went down. It went down precipitously. Instead of killing almost every rabbit that infected, it would kill fewer and fewer, and the virulence went down, and also the rabbits became resistant. This is all about virulence evolution. Let's think about the two scenarios. First, 1950s, a perfect storm, just like the 1918 flu. Tons of rabbits, all of them were uninfected. They had never seen the myxoma virus before. Extremely easy transmission, these very crowded populations of rabbits, many new hosts available, essentially selecting for extremely high virulence in the pathogen. But by the 70s, almost all the rabbits either were dead or were already infected and thus resistant, so hosts were extremely rare. Transmission was slow because it was very difficult to find an uninfected host, and the host had evolved an immune response. So all these things selected for viruses that were much less harmful. Okay, so if you've fallen asleep for chapter 2, here's the main points reviewed. Pathogen virulence, the harm a pathogen cause a host, it can change, it can evolve, and this can occur pretty rapidly. Virulence shifts, whether a pathogen becomes more or less harmful, they can be predicted by this theory. And the main theory says if transmission is easy, if there's multiple infections, this should favor high virulence. If there's only single infections, if transmission is slow, or if there's some kind of immune response, this should select for lowered virulence. Okay? So now you've learned some of this theory. Now what's nice about this theory is that it applies very well to any kind of infection. It doesn't have to be an infection that's harmful. It could be an infection that's beneficial. And we can use this theory to make some predictions about why a symbiont might also be harmful or helpful. All right, so now we'll move on to some of my research, and I'll tell you about some ideas that we've tested in the lab to try to understand why symbionts can be harmful or helpful. And I'll be telling you about this in the bean system that I study. So we'll be addressing 3 fairly simple questions. 1, where do the harmful guys come from in the first place? Question 2, how common are the bad symbionts? This will be really important. And finally, the most important question is, how can hosts respond? Okay, so again, a quick reminder of the system. The system are bean plants. There's about 10,000 different species of bean plants that are infected with these bacteria. And why I choose the system is that it's very easy for experiments. I can grow up thousands of bean plants under sterile conditions. I can infect them with lots of different strains of bacteria. And I can study their generations relatively quickly. Point two that's important is that these bacteria are very important for our crops. Bean plants, of course, are crops that we eat ourselves. Potentially more important, they're some of the main crops that we feed to the animals that we eat. They're important fodder, including clover and alfalfa. And in the future, they're probably going to be an important biofuel. So understanding how the symbiosis can be maintained and maximized is going to be very important for humanity. Finally, these bacteria, although they are symbionts and they live in the soil, they have some important medical relevance since they bear a lot of similarities to some pathogens, and they're actually closely related to a lot of animal pathogens. All right, a quick tour of how infection occurs. Of course, as I said before, beans begin life symbiont-free. Here's a little seed sitting in the soil. And of course, that soil is populated by these bacteria called rhizobia. And the seedling starts germinating. It starts sending down roots. And before that seedling has even sprouted its first leaf, it's already infected by these bacteria. And the bacteria form these little nodules, these little tumors on the roots. And as the plant grows, the bacteria grow within those nodules. But then when the plant's life is done, once it's flowered and fruited and starts drying up, what it does is it releases those symbionts. They get released back into the soil, and then they can start again the next season. All right, what about the harmful guys? Where do they come from? Now remember, this is a symbiosis. This is a cooperative system. It's based on this nice trade. And if we want to think about it, we can think about it in terms of economics. Here's the plant, the legume. It's gathering energy from the sun. It's photosynthesizing. It gets a lot of sugars by combining carbon dioxide and water. And it gives a subset of its sugars to those bacteria. Here's a little streak of bacteria sitting on a petri dish. Here's literally a strain that I took from this plant. Of course, I told you that the bacteria are giving nitrogen, this key resource, back to the plant. Everyone's happy. It's a good trade. Why would you want to be harmful when the system is so cooperative? Well, here's why. The system is a little bit more complicated. Remember I said there's multiple infections. This is a key part. So let's look at the roots of this plant. Here are the roots right up here. And remember, there's multiple different tumors. Each tumor carries a different strain of bacteria. Here's literally the strains of bacteria that are cultured from each of those tumors. And each bacterial strain will vary. So maybe this strain, it's really cooperative. It's giving the plant lots of nitrogen. This strain, very cooperative, giving the plant lots of nitrogen. Well, what about this guy? He's barely giving it any nitrogen at all. So if we think about this in terms of economics, this strain is buying low and selling high. It's bearing all sorts of profits from the plant, and it's giving little to nothing back. So one idea that's been suggested is that because this guy is getting so much more and giving so much less, it should really be able to profit. This should be the best strategy. The best strategy is don't be so generous, be a cheater, be a freeloader. So that's the theory where harmful symbionts come from. Now that's based on a lot of mathematics and equations that I've skipped, and what we want to do is find out, is this really true? Does the theory hold up? So that's the idea. Well, now we want to actually go out into nature and look for harmful symbionts and find out, are they common? Are they out there? How common are they? So here's where we go out back into nature, and we go out and gather some plants. Again, here's the plant that I study, Lotus strigosus. What we do is go out and we collect a bunch of different plants, and we culture the bacteria from those plants. So here are some of the places that I've gone. This is the coast of Northern California. This is Sonoma County. This is Bodega Marine Reserve. And here are some field sites closer to home. Here's UCR's campus right here. Of course, this is in the springtime when things are nice and green. And here's a hillside. Here's, again, the beautiful UCR campus, the San Gabriel's. And we've literally collected some Lotus strigosus right here in this hillside. So I've got a field site that's only 5 minutes from my lab, which is very convenient for me. And then we bring these plants back into the lab. And of course, the first thing that we do is we isolate the nodules. We pull off those tiny little tumors. And we culture them. We culture them because we're not really interested in the plant so much. We're interested in the bacteria. This particular strain is called Bradyrhizobium japonicum. And we take those bacteria and we culture them. We always want to culture them, grow them on petri dish, and then we want to archive them in the freezer. So we have a freezer literally with thousands of different strains. So that way, if we find an interesting bacteria, we can always bring it back to life, pull it out of the freezer, and study it some more. Now, we can't just culture the bacteria. What we need to do is study their DNA as well. We need to sequence some of the DNA of the bacteria, and we build evolutionary trees of the different strains. Now, what this does is it tells us which strains are different from each other, and this is really important. If we are picking up all these different plants and culturing all these different strains, one thing we'd like to know is whether we're just culturing the same exact strain over and over again. And when we want to find out in a particular plant with these 5 tumors, is it multiply infected? Does it have a lot of different strains? We want to be able to sort out the good guys and the bad guys. So the sequencing basically gives us a DNA fingerprint that allows us to identify any particular bacteria. It's got a DNA fingerprint because just looking at the bacteria is not very informative. So that DNA fingerprint is kind of like its ID. All right, so once we've given all these strains IDs, we can build these evolutionary trees. And we can actually learn a lot just by doing that, just by gathering bacteria and doing some sequencing. Here's some important things that we can learn. One, just like many of the pathogens that have been studied, these bacterial infections are extremely diverse. There are many different kinds of strains. So we could look at an evolutionary tree of influenza, and it might look just like this. So this epidemic in the plants, it's a good epidemic, but the genetics are very similar. Fact 2. What we find is that some strains are extremely abundant and extremely widespread. So sometimes we'll find the ID of a strain, and we'll find that strain over vast space. So we'll find it over several kilometers that that strain has spread. This is important because it tells us that transmission in the system is relatively easy. A strain can spread across a lot of space quickly. Another important fact. What we found by IDing the strains genetically, we found that all hosts are multiply infected. Every single time, even the tiniest plant with only 5 infections, it's usually going to have 3, 4, or often 5 completely different infections. Each nodule, each little tumor, harbors a different strain of bacteria. Okay, but when we look at all those strains, now what we want to do is find out what are the good guys and what are the bad guys. So here's where we go back to our freezer, we grow up those different strains that now all have genetic IDs, and we become little farmers. We infect them into plants. Now we don't just do normal infections. What we have to do is grow plants under sterile conditions, and we have to sterilize everything that they're going to touch, because we want them to dig into soil that has no bacteria at all. We're going to supply them with bacteria. So we have these little pots, and every pot is numbered. And although you probably can't see, they're very tiny baby plants that are starting off life. And what we do in these greenhouse experiments is that we give plants a single infection, and we compare that to a control plant that has no infection. So all we want to do for the bacteria is find out, does it make the plant grow more, or does it make the plant grow less than having no bacteria at all? It's a very simple experiment. So here are some results. Instead of giving you a graph, I'll just show you what some of the results look like. Here's strain 24, one of my favorites. It makes the plants grow like crazy. It's a very beneficial strain. If I was a farmer, this is a strain I'd want to use. I'd want to use that unless I discovered strain 49, which I found later. It's even more beneficial. These plants have only grown for 8 weeks. They're huge. Here's some control plants. Those are plants that get no bacteria at all. They get no fertilizer. They're pretty sick. They're barely alive. And then here's famous strain 2. These guys are infected with bacteria, but if you can see them, you can see that they're very small. This strain literally causes the plants to shrink. Okay, so some of the infections are good, and some of them are bad. Here's what a good infection looks like. When we pull out the plant from the soil and we look at the infections, here's the plant up at the top. It's nice and big. Remember, it only gets to live 8 weeks before we mercilessly pull it out of the soil. Here's some of the infections. The nodules are very big. So that suggests that the infection is going well. Here's another plant, different kind of infection, less beneficial strain. The plant hasn't grown as much, and the nodules are a little bit smaller, but still it's beneficial. Now here's that strain that made the plants shrink. This is starting to look like those harmful strains I was talking about. The plants are barely alive. They literally have shrunk over time, and you can't even see the nodules. You can't even see them. They're extremely tiny. But what's interesting is that they're spread all over the plant's roots. This strain is causing almost like cancerous growth on this plant. They're spread across the roots, and they're causing many very tiny infections. So here's some graphical representations of the plant's growth. Here's a couple different strains of bacteria. And these bars show how much the plant grew. Here's 0 over here. So that means 0 growth. So these are 4 beneficial strains that made the plants grow very well. And here are some strains that aren't so beneficial. Here's that strain 2. The point is below 0. So that means it's making those plants shrink. But what we really want to know is what's going on with the bacteria. Remember, we had the economic theory that if a bacteria is a freeloader, if it's cheating the hosts, it should be able to do even better than the beneficial strains. So here's the growth of the bacteria. We open up those nodules, and we look at the proliferation of the bacteria. And there is strain 2. So strain 2 is causing terrible harm to the hosts. The hosts are shrinking, and this bacterial growth is off the charts. So it supports the economic theory. This bacteria is literally buying low and selling high. It's growing like cancer inside the host. It's a symbiont, but it's causing harm, and it's apparently profiting from that harm. All right. So what we found is that most of the strains were good. We found a couple strains that were marginal, and we found one really bad strain. It's pretty consistent with what's been found in other systems. All right. Finally, what we want to know is what can we do? In this case, it's not really what can we do, but what can a bean plant do? And what's nice, and why I've introduced you to all these different systems, is that there are some general rules about symbioses and also some general predictions. These predictions work whether you're a coral, whether you're a human, whether you're a tree or a plant. There are 2 main predictions about how hosts can control infections. Prediction 1 is you stop the bad infections even before they start. So this is the idea is as you're picking up the infections, you have some detection method, and you could stop the bad strains even before they take hold. So that would be a pre-infection hypothesis. The other hypothesis is a post-infection hypothesis. This is the idea that you get a bunch of different infections, and while you're infected, you can somehow modulate them, maybe give the good infections reward, or maybe punish the bad infections. So we're going to test both of these ideas in our tiny little bean plants. And here's what we do. So before we had a simple experiment. Plants were either infected with bacteria or infected just with water at control. But now we're doing what I call a 3-strain experiment. So we've got 3 different strains of bacteria. We've got a beneficial strain, coded in blue, we infected into 6 little plants. Here's another beneficial strain infected into 6 plants. Here's our pathogenic or cheater strain. It's infected into 6 plants. And then control, which of course is just water. So that's similar to our experiment before, but here we spice it up by having a couple co-infections. And what we really want to know is, remember, plants are always multiply infected. We want to know when they're digging their roots into the soil and they've got a good guy and a bad guy to choose from, can they choose the good guy? And we're going to do this with multiple different strains. So this whole experiment is replicated with lots of different kinds of strains, but this is the basic unit of the experiment that's replicated with lots of different combinations. Here's some of the things we measure: plant growth, number of nodules, how big are those nodules. But we also look in those nodules and figure out how fast are those bacteria growing. Are the bad guys winning or are they losing? All right, first we'll test the pre-infection hypothesis. All right, so now we're going to look at some data. These are data that are growth rates of plants with different strains in the 3-strain experiment. So some strains will start to look familiar. Here's strain 2 again, and it's causing 0 growth. Those plants are not growing. Here's a couple beneficial strains, and what these letters tell us is that those beneficial strains are significantly better than 2. All right, so here's 2. It's going against 2 very beneficial strains. Here's 2 again, competing against 2 very beneficial strains. And then here are other competition experiments with less variability. All right, so that's just the effects on the plants. Now we want to know, Are the good infections getting in, and are the bad infections being stopped? So now we're going to look at the identity of bacteria that actually get into the plant and how often they get in. And what we see here is here's the beneficial strains, and on the left axis is the infection rate, and it goes from 0 to 1. So strain 18, it's beneficial, its infection rate is 1. It always was accepted by the plant. Strain 24, beneficial, its infection rate is 1. But the harmful strain, it was stopped. It wasn't stopped all the time, but it was reduced. Next set of experiments, same thing. The beneficial strains are always accepted by the plant. The harmful strains are slowed down, not completely. Here's more experiments. This experiment, all the strains were about the same, and the plant didn't even notice the difference. It accepted them all. Okay? So what we see is a pattern where a plant has a system. It's an imperfect system, but a system where it can slow down the bad infections and stop them some of the time, but not all the time. The good infections are always accepted. So we can look at this in a larger pattern by making a graph that looks at the relationship between how beneficial a strain is and its likelihood of getting in a plant. So what we're looking for is a positive relationship. What that means is the more beneficial you are as a plant, as a bacteria, how much you make the plant grow should give you a better chance of getting into the plant. So here, our most beneficial strain is up here. It makes the plants grow quite a bit, and its infection rate is 1. It's always accepted. Here are some of our most harmful strains. They basically cause the plants to shrink, and they're almost never accepted. Okay, so that supports the hypothesis pretty well. All right, let's next test the second hypothesis. Once the plants are infected, can they slow down the bad guys? Now, we know they can't stop the bad guys and infection completely, so we want to see, can they slow them down? All right, so here are those same graphs again, the growth rates. Remember, just reminds us that some strains are beneficial, some strains are harmful, and we want to look within those nodules and see which strains are winning. So now, instead of an infection rate, we're literally counting bacteria. We're counting the number of bacteria within a plant by culturing and then extrapolating. So here we can look at population size. All our graphs are logarithmic, so it's exponential. And we can see that here, the beneficial strains, there's about 100 million of the good strain. The bad strains, there's about 10 million. So it's not a perfect system, but the plants are slowing down the bad strains. Here's a more striking example. Good strains, there's about 1 billion bacteria per plant. Bad strains, there's about 1 million. So almost a 1,000-fold difference. We basically got the same results as before. The plants were able to detect the bad strains and slow them down. When we look at the relationship between how beneficial a strain is and how rapidly it can proliferate in a co-infected plant, we find the same pattern. We find the pattern that the strains that are most beneficial are also allowed to proliferate the most. Here's a population size of about 1 billion bacteria per plant. Most harmful strains have a population size that's about 100 to 1,000 times lower. So those plants are pretty smart, and they're doing a good job in choosing their symbionts. All right, so if you fell asleep during part 3, here's our some conclusions again. These bean plants, just like most hosts, they harbor diverse strains of bacteria called rhizobia. Most of these strains are beneficial. They make the plants grow like crazy. They're really good. We did find some strains that are freeloaders. We found one strain that's really a pathogen, very harmful. But what we also found is that there's some good news. Both ideas about what hosts can do, we were able to find. They're imperfect systems, but we found that the plants can stop infections of the bad guys. They can't do it perfectly, but it's pretty good. But they can also slow down the bad infections once they occur. Now, you may have come here with the idea that you really wanted to learn about what we can do about our own symbionts, and it would be great to find some people to talk about that. But what they'd be able to tell you is a lot about the genetics of these bacteria and really not much more. We really don't know that much more about the bacteria that are living in our guts. And one problem is that it's extremely hard to study them. As I said, there's hundreds of species, and you could simply not set up an experiment where you'd infect thousands of babies with different strains of bacteria like I have done with my baby bean plants. Obviously, there are some ethical dilemmas. All right, so I've not done this work alone, so I have a lot of people to thank. First of all, I'd like to thank UC Riverside first for the opportunity to give this talk, but also for some funding I've gotten. A lot of this work has been funded by the National Science Foundation, which funds basic research, but also funded by National Institute of Health, which funds medical research. Here are some of my collaborators. Collaborators at Berkeley, Oregon, my postdoc here at UC Riverside, and some other collaborators back at Berkeley. But I'd also like to thank all the members of my lab that helped with some of the experiments that I've described to you today. And finally, I'd like to thank my daughter, Nora, for posing for a lot of the pictures and also being very patient with me as I spent a lot of time thinking about what I was going to talk to you about tonight. All right, so I'm done, and I'd be happy to answer any of your questions.
No, and the hot zone, the hot zone and the Ebola bacteria, and you bleed out, and it finds its host immediately, and you live about 48 hours. You never read that book?
No, I have not read that book. It sounds scary, though.
Well, it was. So, I mean, for instance, the swine flu. I mean, I heard this morning on the news that they're now saying that it's not a pandemic, and that it's really not it's not spreading as rapidly as they expected it to, because it's not finding the new hosts.
Yeah. Well, all I would say is that any information you hear about the swine flu, I'd be very skeptical. I'd be skeptical because it's happening very quickly, and basically we just don't know that much about how fast it's spreading. And whether you're defining it as an epidemic, which is isolated to one part of the planet, or a pandemic, meaning it's everywhere, it's somewhat irrelevant. What's important is how much harm does it cause? Obviously there are pandemics of rhinovirus that cause no harm at all, and they're irrelevant. They don't. So it's not necessarily how common it is, but how harmful. And the problem is that you can have pandemics, and there's a lot of people that are probably infected with H1N1 that are asymptomatic, and those are simply data that we don't know anything about. So it's really the very harmful strains and the harmful conditions that we need to worry about, and that's where getting the most important people vaccinated is the critical thing, and hopefully that will occur before anything more harmful happens.
So I didn't understand in your experiment when you were trying to figure out whether the plant could stop bacteria from getting in. How did you tell it was the plant that was stopping the bacteria rather than the strains differing in their ability to get into the plant?
It's an excellent question. So the question is, and this is a problem in any kind of symbiosis, I had the hypothesis that the plant was controlling the strains, and that the plant essentially has some kind of detection method to stop the bad guys and select for the good guys. But there's another way that this system could also be working. It could be based upon competition among the two different strains of bacteria. And it could be that the more cooperative strain is also more competitive. Maybe it's able to grow faster, or maybe it's able to push away the bad bacteria. Now, this is not a hypothesis that we can completely rule out, but we do have a couple pieces of information. One is we do in vitro experiments in which we compete the bacteria against each other. So one thing we want to know is it just that the good bacteria that the plants are selecting are able to outgrow the bad ones? We also test hypotheses like, are the good bacteria able to get onto the plant roots faster? And when we do these experiments, we basically find no patterns. And this is something that's been supported by studies in agriculture. For agricultural biologists, what they'd love to find is a strain that's incredibly beneficial and incredibly competitive in the soil and would always get to the plants. But it never works because of economics. There's always freeloaders, and whenever they infect bean plants with these strains, there's always evolution. There's always evolution that's going to favor the selfish strains. So inoculating bean plants with these bacteria is always an unstable proposition. That's a great question, though.
Well, do I need you to use the microphone, or can I put like that? I know that I won't use a microphone. Can I talk like this?
Yeah, I can hear you.
Now I know that when sometimes when a host is infected, capable and immune response, that when so even when there is a lot of bacteria getting infected, there's still another what percentage that's not being infected. So the percentage that's not being infected become high alert status in the immune system. But I thought that the infection has nothing to do with them. It's they were just not infected. Like the group was infected, group A was not infected. Group A should have the most immune response because they were not infected. It has nothing to do with them.
Okay. So yeah, so the question is trying to relate the immune response and how that's going to affect the pathogen, whether it's a virus or bacteria or anything like that. So the subpopulation that's not infected, that the pathogen has not hit, that's not part of the equation at all. Those are hosts that are potentially available for infection, but that's not going to shape the evolution of the pathogen. What can shape the evolution of the pathogen is the part of the population that evolves resistance. So remember, when we're looking through the eye of the pathogen and trying to understand, all that matters to it is whether it can find a new host. So if it finds a new host and that host is susceptible, then that pathogen can travel to that host. If it finds another host that's resistant, it cannot infect that host is not available to it, that host basically doesn't count. So the immune response, if the immune response is selective and it's only going to affect strains that are more harmful, then those strains will be selected against. Okay, any other questions? Way in the back.
How evolutionarily distant are your pathogenic strains from your beneficial strains?
It's a,
Close relatives that behave differently.
Yeah, that's a great question. So the question was, how closely related are the beneficial strains from the harmful strains? So I only gave a part of the story. There's a lot of other complexities to it, too. One thing that we found is that we find tons of strains that are closely related to the good guys and the bad guys that can't even infect plants at all. They can't form the tumors. They're ineffective completely. And what we found is that the harmful strain was actually closely related to those guys. And what we also found is we're really interested in the genetics of the system. And one thing that is interesting about these bacteria is that all the traits that allow the bacteria to infect the plant are harbored on a special part of the bacterial's genome that can travel. It can move from bacteria to bacteria. It's kind of like a traveling genetic element. And what we found is that this harmful strain has picked up a bunch of new genes from something else. And we actually don't know who those genes are related to because they're not related closely to anything that we found in our population. So in terms of the symbiotic genes, it's very distantly related. In terms of the other genes, it's very similar, but similar to these strains that can't even infect plants. So that aspect of it is pretty confusing to us.
Just a quick question. How does this research explain the emergence of new species, the symbiosis? How does it explain that?
Okay, the question is, how does this research explain evolution of new species or evolution of symbioses? It doesn't really at all. So there's really two kinds of aspects when we look at an evolution. And one is the maintenance of a system, how a system can kind of change, maybe modulated up or down. So we can understand the symbiosis, how these bacteria can evolve to be more or less beneficial. But then there's another question is, how does the system originate? Where do these symbionts come from in the first place? And that's a completely different question, and it's actually a much more difficult question to understand. How did a bacteria come to infect a plant? These bacteria are very ancient. They've lived on the earth before plants ever existed. How did they evolve traits that allow them to infect, invade plant tissue, cause a new organ on a plant? And that's an excellent question. And as in most sciences, evolutionary biology has lots of open questions that are a complete mystery, and that one is relatively unexplored. That's a great question.
How did you control variation in the hosts so that you know that the results that you got were due to a variation in a strain versus a variation in the host?
Okay, so this question is saying, how can I study? What I'm doing is infecting a lot of different hosts with different strains of bacteria, but these hosts also vary. These hosts are not all genetically identical. And that's a great question. There's a couple different ways you could do that. One is you could infect hosts that are all really closely related to each other, and thus there would be less genetic variation coming from the host. Or you can infect a lot of different hosts that are distantly related from each other and hope that you kind of lose that variation in the wash. And we took the second approach. The problem is that there's quite a bit of variation in the host, and one thing that we were worried about is that some strains might be slightly more beneficial in some hosts and slightly less beneficial in other hosts. And this is the case, and we can actually quantify some of this variation as well. So what we do is we try to design experiments that are big enough that the variation caused by the bacteria will kind of swamp out the effect of the variation caused by the host. And sometimes we can do this, and sometimes we can't. But what's important is that the growth effects of the bacteria are pretty massive, and the effects caused by the host variation is relatively small. Okay, this is a question down here.
Yes. Is it possible to say that your legumes have an immune system that can be compared to a human immune system in any sense?
Okay, so the question is, can we compare the immune system of a plant to immune system of an animal, for example, like humans? And I would say no, because one thing so you can describe an immune system as any kind of reaction that's differently affected by different microbes. You know, maybe stopping some microbes and allowing some microbes in. Basically, whether you can recognize foreign from yourself. Now, our immune system is adaptive in that it's always building up new antibodies to new antigens and remembering those and saving those antibodies for a later infection. So that adaptive aspect is something that is not found in any plants as far as anybody has known. They just have this innate system of saying, all right, you're a good guy, you're a bad guy, and reacting to those differently. But there's no kind of memory that's built up like our adaptive immune system. Okay, any other questions?
So I guess the take-home message, if we're feeling lonely, we shouldn't be.
Yeah, that's a great ending point.
So I did remember what else I was supposed to do. One thing is I was supposed to recognize any teachers who might be here. I understand the numbers are pretty small because there's a teacher award ceremony that was scheduled for tonight, and there are many people at that. But if there are any teachers who were able to come, or members of the school board, here we are. So thank you for coming very, very much, Lou. And the last group I would like to recognize are the members of our Science Circle, a group that has actually supported this lecture series and to whom I am extremely grateful. Would you please stand up and be recognized, please? And finally, to Joel, I want to thank you very much. A very, very interesting and exciting talk. This business about symbiosis and how it develops is, I think, one of the really great questions. Hopefully, I'll live long enough to know some of the answers of it. But thank you very much, Joel. And thank you all so much for coming. See you next spring.